Okay, good morning everyone. Today I will be doing my presentation on carbohydrates for my chemistry class. My name is Sabrina Munoz. We will start off with what is carbohydrates. Next slide. Carbohydrates can be defined chemically as neutrally compounds that includes carbon, hydrogen, and oxygen. Carbohydrates can come in simple forms and complex forms. The simple forms include sugars, and complex forms are presented as starches and fibers. Now, carbohydrates can also be classified into mono, di, tri, poly, and oligosaccharides. The smallest carbohydrates are monosaccharides such as glucose, whereas polysaccharides such as starches and cellulose are large, and when you talk about the length, it can be indeterminate. Next slide. So now we will talk about how plants use specific compounds to create carbohydrates for them to use as energy sources. And this is basically photosynthesis. Next slide. So here we have photosynthesis, and this is the equation for photosynthesis, which is carbon dioxide plus water. By using light, you make glucose and oxygen. Now, within this entire process of photosynthesis, we will be focusing on the Calvin cycle, because the Calvin cycle is where carbon dioxide is fixed into sugars, which is glucose that the plant uses. And now the steps for the Calvin cycle is carbon dioxide reduction, carbohydrate formation, and regeneration. Next slide. So carbon fixation is the first step. Here we have carbon dioxide that is reacted to produce glyceraldehyde 3-phosphate, which is G3P, and the enzyme Rubisco catalyzes the carboxylation of a 5-carbon compound to make a 6-carbon compound. This 6-carbon compound will then split into 2 carbon molecules, which is the 3-PGA. So after this, we have the reduction phase. And the reduction phase is where 3-PGA is converted into G3P with the use of ATP and NADPH. So after the reduction phase, here we have the carbohydrate formation, and this is basically where the G3P is turned into the carbohydrates that the plant will use. Finally, we have here where the regeneration phase, and the regeneration phase is basically where the G3P plus the ATP will be used to make more Rubisco in order for the cycle to begin all over again, so that we can make more glucose for the plant. Next slide. Now the next process that we will be talking about is respiration, specifically for plants. Next slide. Okay, so here we have the same picture on the previous slides, but on the bottom we have the equation for respiration. And when you really think about it, the equation for respiration is the equation for photosynthesis, but in reverse, because here we have glucose of the plant plus the oxygen, which will make carbon dioxide plus water plus ATP as byproducts. So here we just have in word form the components of the respiration. Glucose plus water will give you carbon dioxide and water, glucose plus oxygen. So the breaking of carbon bonds of the complex compounds through the oxidation within the cell will lead to energy that will be used by the plant, and that is basically what respiration is. Now, when we talk about respiration specifically in plants, roots, leaves, and stems are all responsible for their own section of respiration. Next slide. So when we talk about aerobic and anaerobic types of respiration, they have a common series of initial steps termed glycolysis. And during glycolysis, carbohydrates are converted into pyruvic acid, and the pyruvic acid will then enter into the mitochondria, where oxygen and necessary enzymes are available. The pyruvic acid is finally converted into carbon dioxide and water, and this basically constitutes the Krebs cycle, which is the important part of respiration. So here we will continue with carbohydrates, since we already understand the process of respiration and photosynthesis in plants. So the general empirical structure for carbohydrates is CH2O and then close brackets with an N on the bottom. Now this is very important because we learned that carbohydrate is made up of carbon, hydrogen, and oxygen. Now the functional groups when naming carbohydrates, my bad, are ketose and aldose. Now the ketose and aldose are the functional groups. So how to determine if it is an aldose or a ketose? You look at the carbonyl group. In an aldose, the carbonyl group is located towards the end of the chain structure, and within a ketose, the carbonyl group is located within the middle of the structure. And examples of ketoses are fructose and ribulose, and examples of aldoses are glucose and ribose. Next slide. So another very important factor that you need to take into consideration when you're naming monosaccharides or carbohydrates on a whole is the D and L sugars, because you want to know, this basically refers to the location of the OH on the chain structure, because you want to give the reader an idea on where the OH is placed. So if the OH is on the bottom chiral center, if it points to the right side, the sugar is a D. So here, this example, this is a D sugar because the OH is on this side. Now if the OH would have been on the left side, the sugar would be classified as an L. So this is a D-glyceraldehyde. And we know that this is a glyceraldehyde because a glyceraldehyde has 3 carbons, making it a triose. And that is another important factor when we talk about naming carbohydrates. So what is a chiral center? A chiral carbon is a carbon that has 4 different groups attached to it. So the chiral carbon in glyceraldehyde is the middle carbon, which is this carbon. And when we talk about D-glyceraldehyde and L-glyceraldehyde, D-glyceraldehyde can be found in nature naturally, and usually L forms of sugars are synthesized within the labs. And then here is just explaining the chiral center and how if the OH on the bottom chiral center points to the right, it is a D, and it just repeats exactly what I just explained. Next slide. So now we will move on to monosaccharides. What are monosaccharides and how to name them? So monosaccharides are the most basic form of carbohydrates. And when we want to name a monosaccharide, it's very straightforward because we already went over some of the steps. First, you look at the functional group, if it is a ketose or an aldose. And we know that a ketose and an aldose is just knowing the location of the carbonyl group. Then the next step is the number of carbon atoms in the main skeleton. If it's 3 carbon, 4 carbon, 5 carbon, or 6 carbon, we have different names. We have triose, tetros, pentos, hexos. Now you just combine both systems to give even more information to the reader and a name to the structure. So here we have an aldopentos. So we know that aldo because it is an aldehyde, and pentos because it has 5 carbons. So when we talk about glyceraldehyde, we know that it is aldose, and we know that it is an aldehyde. Here we have other examples, aldohexose, we have ketopentos, and we have ketohexose, which are just identifying the location if it is a ketose or an aldose and the number of carbons. Next slide. So important monosaccharides to remember are glucose, fructose, and galactose. So the first one that we're going to be talking about is glucose. So glucose, fructose, and galactose have the same chemical formula, which is C6H12O6, but they differ in the organization of atoms. Therefore they are isomers. So glucose is one of the most common monosaccharides in nature, used by nearly every form of life. It provides both energy and structure to many organisms, and it is a 6-membered ring. Glucose molecules can be broken down via glycolysis, which will then provide energy and precursor for cellular respiration. Now, if a cell does not need any more energy, it can store this glucose, and you can then use it whenever the plant or the animal needs more energy. So plants produce glucose as cellulose, and this is an aldose. So right here, this is a cyclic form of glucose. You just see the bonds, like when we would want to change the open chain into a ring structure, this hydrogen will move up here to the first carbon, and this double bond will break, which will then form a OH group, which is this, and then this hydrogen will remain. So the only thing actually that changes is that this bond will break because this hydrogen and this oxygen will bond and form OH. And then this is how we will have the ring structure. So another important thing to remember is that there's the alpha and there's the beta form. The alpha form is when the OH is towards the bottom, and the beta form is when the OH is towards the top. And this ring is a pyranose ring because it is a 6-carbon ring. If it would have been a 5-carbon ring, which we will talk about in the next slide, it would be known as a furanose ring form. Next slide. So the next monosaccharide is fructose. So fructose, although it is almost identical to glucose, it is slightly different within the molecule because the formula is CH2O6, which is the same, but the structure is much different. So fructose is a ketose when, go back, where glucose was an aldose. Okay, change slides. And this is a 5-carbon membered ring. So when we have the ring like this, it will be known as a furanose instead of a pyranose. So fructose is a natural sugar that is present in fruits, fruit juices, certain vegetables, and honey. And here we just have explaining or pointing out if it's an alpha or if it's a beta ring. So the next one is galactose. Galactose is produced mainly in mammals. Mammals use galactose in milk to give energy to their offspring. And galactose is combined with glucose to form the disaccharide lactose. So galactose is an aldohexose, and a very important fact is that glucose and galactose are stereoisomers. This means that they have the same chemical formula, the same functional group, but the only difference is the position of the OH. So that's basically the definition of a stereoisomer. And here we see the ring structure of the galactose, and here, because the OH is on the bottom, we know that it is in an alpha form. And then this just is the chain structure to the ring structure. And then we have ribose. Ribose is a 5-carbon sugar that makes a 5-membered ring, which is a furanose. And ribose is found in nucleic acid in every human being on the planet within RNA. Well, actually, no, I'm sorry, we have DNA, but ribose is found in RNA. And this is just the open chain cycle, and then these are the rings. This is the ring for ribose, and this is deoxyribose. Next slide. So when we talk about deoxyribose, it is a 5-carbon sugar molecule that helps form the phosphate backbone of DNA molecules. Now, we have ribose and we have deoxyribose. And the only difference between ribose and deoxyribose is the presence and the absence of the oxygen. So that is one of the major differences when we talk about DNA and when we talk about RNA. Now, we as humans, we have DNA. Next slide. Now, we're going to test for carbohydrates. So there are a couple of tests that you can do to test for carbohydrates, but the first one that we will be talking about is a tolerance test. So this test tests for aldehydes, and the concept is that the color change will be used to determine if the substrate that we are testing has an aldehyde or a carbohydrate or does not. So we will be using, this is the reagent, and this is diamine silver 1 complex with the presence of OH. Now, the concept behind it is that the silver ions within the reagent are the oxidizing agent, and these ions are reduced to silver metal. Now, the silver then deposits as a mirror on the sides of the reactant vessel. Therefore, if the sample is an aldehyde, a positive result will be obtained, and the test tube will turn silver. And that is how we will know that there is a presence of aldehyde within the substrate that we are testing. Next slide. So the other test that we can do to test for carbohydrates is the Benedict's test. Now, this is actually split into 2 parts that I will be explaining. The first part is the reducing sugar, and the second part is for non-reducing sugars. And it's very straightforward. So we know that it is used to detect the presence of reducing sugars. And the reducing sugars, if you don't know, are monosaccharides and disaccharides, which are maltose and lactose. Now the reagent for the Benedict's test is copper 2 sulfate. And the whole idea or concept behind it is that if the presence of reducing sugar are within the substrate that we are testing, we will know because of the color change. And the process is really easy. You get your substrate that you are testing and you add the copper 2 sulfate, and then you get the test tube and you insert it into a water bath for 5 to 7 minutes with temperatures being 100 degrees Celsius. Then you take it out and the color should change if there is a presence of a reducing sugar from green to yellow to orange to red, depending on the severity or the concentration of reducing sugars within the substrate that you are testing. Next slide. Now, if the process is done, the test is done, and there are no changes, that could mean two things. The first thing is that there are no sugars present, or the second thing is that there are disaccharides present, and therefore the test is not able to detect it. Disaccharides include sucrose, and sucrose are non-reducing sugars, therefore they are not affected by the test. Therefore, there are some preliminary steps that need to be taken in order for the disaccharides to be detected when doing the test. So the first preliminary step is you add hydrochloric acid to the sample, and then you add it in the water, the boiling water, for 5 minutes, and this is so that you can neutralize the sodium hydrogen carbonate solution. So after you neutralize it, you go ahead and you proceed with the Benedict's test for reducing sugars. And the hydrolysis of the non-reducing disaccharides is basically breaking the disaccharides into two monosaccharides, because a monosaccharide is 1 and a disaccharide is 2. So testing for reducing sugar means that we are testing for monosaccharides. And if there is a presence of a disaccharide, we cannot detect it. So that's why we have to break it down into two monosaccharides. Next slide. So now we will talk about disaccharides. So what is a disaccharide? A disaccharide is formed by joining two monosaccharides in a condensation reaction, and within a condensation reaction, water is a byproduct. So disaccharides are sweet tasting and soluble carbohydrates. An example are maltose and lactose. So disaccharides are made via glycosylic bonds. And this picture, as you can see right here, is an example of a maltose. And a maltose has a 1,4 glycosylic bond. I will talk about this more when we move into naming and just explaining other types of disaccharides. So a couple steps to remember or to take when naming them is 1, the type of atom involved in the linkage, being it oxygen or nitrogen, and then the configuration of the anomeric hydroxyl, which we also talked about in monosaccharides, if it's alpha or beta. Then you name the first monosaccharide, the root name, followed by pyranosyl, which is a 6-ring, or furanosyl if it's a 5-ring. And in monosaccharides, we were furanose and pyranose, but here in disaccharides is obviously slightly different. Then the first sugar, then the second sugar, and then the configuration of the anomeric hydroxyl of the second sugar, and finally name of the second monosaccharide, the root name, followed by pyranose. So it's a couple of repetitive steps, but it's very handy, and I will be giving example further within the slides. Next slide. So important disaccharides are maltose, sucrose, and lactose. First, we will be talking about lactose. So lactose, because it is a disaccharide, they are all made up of monosaccharides. So maltose is composed of alpha glucose plus an alpha glucose with a 1,4 glycosylic bond. Maltose is found in germinating seeds and can be broken down for energy. Therefore, it's found in plants. Then we have sucrose. Sucrose is an alpha glucose plus a fructose, which has an 1,2 glycosylic bond. Now, sucrose is transported in the phloem to provide sugars in other parts of the plant. Example, table sugar, cane sugar, beet sugar. Next slide. So here I just have pictures of the ring structure for maltose and the ring structure for sucrose. This right here is the glycosylic bond, and this is the glycosylic bond. Now, these are just the individual monosaccharides, which is a glucose and a fructose. Okay, change. And then the last one is lactose, which is alpha glucose plus galactose. And lactose is found in mammalian milk to provide energy for infant mammals. So here, lactose has a 1,4 glycosylic bond. And we know that this is a 1,4 glycosylic bond because when we number the carbons and then we just look at the specific carbons that are connected within the glycosylic bonds. So this is the first carbon because you follow the 3 o'clock rule. You start from here, the first carbon, and then the fourth carbon. Okay, next slide. So the next part of this presentation includes starch. Now starch is composed of amylose and amylopectin. Really similar, but there are specific differences. The major difference between amylose and amylopectin is that amylose is a straight chain polymer, whereas amylopectin is a branched chain polymer. So amylose, an important characteristic to understand is that amylose has 2 ends for amylase enzyme, while amylopectin has 4 ends, this end, here, and here, for a quicker digest using amylase enzyme for respiration. And this is very important to understand because during respiration, when glucose is needed, the enzyme amylase will come and attach itself to both ends and it will digest it because we want the glucose. But in amylopectin, because we have 4 available ends, it will digest slightly faster. Next slide. So here we have cellulose, and cellulose is basically beta glucose arranged horizontally. So these structures are just glucose stacked in different ways with different linkages. And this is an example of cellulose. The last one is glycogen. The glycogen is the major glucose storage carbohydrate in animals. It is highly branched like amylopectin. However, it has even higher numbers of accessible ends. Therefore, it is found in organs like muscles and in the liver because they have higher metabolic rate, and because they're found in animals have a higher metabolic rate because they need more energy than plants. Therefore, animals have glycogen and plants do not. And that is one of the differences between glycogen and amylose and amylopectin. Next slide. And this is my references, and that is the end of my presentation. Thank you.